Now that we've discussed the action potential and saltatory conduction of that action potential down the axon, we can discuss what happens when the action potential arrives at the terminal button. Well, at the terminal button, we have a bunch of synaptic vesicles filled with neurotransmitter. These vesicles are basically bubbles that are made out of the same phospholipid bilayer that the cell membrane is made out of. And this is important because during the process of exocytosis, that is, the process of flushing neurotransmitters into the synaptic cleft, the synaptic vesicle is going to fuse with the cell membrane. So it's important that the vesicle is made of the same phospholipid bilayer that the cell membrane is made of. Now, when the action potential arrives at the terminal button, it's going to cause the opening of voltage-gated calcium channels. So this is the first and really the only time that we're speaking of calcium channels. And so calcium will flood into the presynaptic cell, into the terminal button, and it will somehow, through a process that's not yet really understood, it will kick off exocytosis. It will cause a synaptic vesicle that is docked to the terminal button. It will cause it to fuse with the terminal button, with the cell membrane, and a small rupture is created in that synaptic vesicle that allows the neurotransmitter to diffuse into the synaptic cleft, all the while maintaining the integrity of the neuron. That is, there's no point at which the inside of the presynaptic neuron is exposed to the outside, yet we are able to flush the neurotransmitter out into the synaptic cleft. And once most of that neurotransmitter has diffused into the synaptic cleft, that synaptic vesicle will undock and be recycled. It will be used to be filled with more neurotransmitter for the next action potential that arrives. So again, the action potential comes from the axon down to the terminal button. It causes synaptic vesicles filled with neurotransmitter to be fused to the presynaptic membrane. A rupture occurs, the neurotransmitter flushes into the cleft, and then we recycle that synaptic vesicle. Now here's an image that gives us a bit more detail. We have, again, the terminal button here. We can even see part of the axon with one myelinated segment here. We see some synaptic vesicles fused to the microtubules, and those are just kind of sitting there waiting for their turn to undergo exocytosis. Meanwhile, there's already some synaptic vesicles docked. The action potential is going to arrive, open these calcium channels. Calcium will cause exocytosis to occur, where that synaptic vesicle fuses with the presynaptic cell membrane and the transmitter is flushed into the synaptic cleft. In this particular example, this is from a video that is discussing acetylcholine. That acetylcholine is going to undergo exocytosis. It will flush into the synaptic cleft. It will diffuse across that synaptic cleft and will bind to transmitter-gated sodium channels in this case. Now we've talked about sodium channels up here in the axon, but those were voltage-gated sodium channels. In order to open those sodium channels, we needed positive voltage. The sodium channels in the dendrites, however, are different in that they are transmitter-gated, not voltage-gated. You could apply all the positive voltage you want to these sodium channels and they won't open up. What they're looking for is a neurotransmitter like acetylcholine to open them up. So the acetylcholine diffuses across the synaptic cleft and binds to receptors on these transmitter-gated sodium channels. The sodium channels open up, and then sodium will follow its concentration and its voltage gradient to the inside of the cell. And this is going to create excitation in the postsynaptic cell. Now, imagine enough sodium comes in that we get enough positive voltage that goes down through the dendrites, through the soma, and all the way to the axon of this postsynaptic neuron, if it's enough positive voltage, it will depolarize voltage-gated sodium channels in the axon, causing the initiation of an action potential. So if you wondered earlier, well, how do we get from resting membrane potential of negative 65 millivolts up to our excitation threshold of negative 55 millivolts? We do that by allowing positively charged sodium to flow into the postsynaptic cell through transmitter-gated sodium channels. Now, what happens once the neurotransmitter acetylcholine has done its job in the postsynaptic cell? It's going to be broken down into its constituent parts by an enzyme called acetylcholine esterase. Acetylcholine esterase. Acetylcholine esterase will diffuse into the synaptic cleft and break apart the acetylcholine into its constituent parts, acetate and choline. So now what we have in the synaptic cleft is not acetylcholine, we have acetate and we have choline unbound to each other. And neither acetate nor choline can open these acetylcholine-gated sodium channels. So we have essentially used enzymatic deactivation to deactivate the acetylcholine. However, there's a different way of enabling or disabling the neurotransmitter and the process of reuptake. So here we have an image from a very similar video, except in this image the synaptic vesicles are filled with a transmitter called GABA. But the same basic thing happens. We have an action potential arrive, which will cause the calcium channels to open, and that calcium will diffuse into the presynaptic terminal button and cause these GABA-filled synaptic vesicles to fuse with the cell membrane. A rupture will occur, and the GABA will be flushed out into the synaptic cleft. The GABA will diffuse across the synaptic cleft to the postsynaptic neuron, where it will bind to receptors on transmitter-gated chloride channels. Now, GABA is always inhibitory, so it makes sense that it's opening chloride channels, because now when chloride opens, chloride's going to follow its concentration gradient to the inside of the cell. Since chloride is negatively charged, that means rather than exciting the postsynaptic cell, rather than depolarizing it toward excitation threshold as we saw in the last slide, this is actually going to hyperpolarize the postsynaptic cell further away from excitation threshold. So it's actually going to inhibit rather than excite the postsynaptic neuron. Now, once we're through using the GABA, in this case, instead of using an enzyme to deactivate the GABA, we're going to use reuptake transporters. These are proteins embedded in the presynaptic cell that can actually grab hold of the neurotransmitter and transport that neurotransmitter back into the presynaptic cell, where it gets packaged in synaptic vesicles and recycled to be used again later on. So again, there's a couple of differences between this slide and the last slide. One difference is that we're talking about GABA, and then we're talking about chloride channels. And instead of enzymatic deactivation, we're talking about reuptake. Now, many of us will be familiar with selective serotonin reuptake inhibitors. These are a form of antidepressant medication that a fair number of individuals use. Selective serotonin reuptake inhibitors. So what this drug is doing is inhibiting these transporter proteins. It's keeping them from reuptaking the neurotransmitter, in this case serotonin rather than GABA. It's inhibiting the reuptake of serotonin into the presynaptic cell so that the serotonin stays in the synaptic cleft for a longer period of time. If it stays in the synaptic cleft for a longer period of time, it has its action on the postsynaptic cell for a longer period of time. The fact that selective serotonin reuptake inhibitors help with depression has given rise to the serotonergic model of depression, the idea that maybe depression results from too little neurotransmitter. Because what happens is when you inhibit these reuptake processes, you not only leave serotonin in the synaptic cleft for a longer period of time, but you cause the presynaptic cell to say, hey, I'm missing some serotonin. I better produce extra serotonin. Okay, so not only are you leaving serotonin in the synaptic cleft for a long period of time, but you're also increasing the amount of serotonin that the brain is making. So this has led to the idea that maybe depression is too little serotonin, and that the reason SSRIs help is because they cause an increase in the production of serotonin. That's a pretty straightforward and reasonable model. However, more recent evidence has suggested that when you increase the amount of serotonin in the brain, you actually decrease the amount of receptors for that serotonin. So that in fact, increased serotonin may actually cause a decrease in the use of serotonin. So it may be just the opposite of what we thought. Rather than depression being too little serotonin, it may be that depression is a function of too much serotonin. And these SSRIs, even though they produce more serotonin, they cause the brain to be less receptive to that serotonin. It's unclear what the correct model on that is, but just a little bit of interesting tidbit there for those of you who are familiar with selective serotonin reuptake inhibitors. Now, we talked about receptors in these postsynaptic cells, and there are basically two types of receptors, ionotropic and metabotropic receptors. The ionotropic receptors are fast, meaning the neurotransmitter binds to the receptor and it causes the immediate opening of a transmitter-gated ion channel. And these tend to mediate behaviors from simple reflexes to cognitive processes. Okay. On the other hand, we have a different kind of receptor called a metabotropic receptor, and it is much slower in its action. Metabotropic receptors are not on ion channels themselves. They are embedded in the cell membrane. They're sometimes called G protein coupled receptors because they're receptors that are coupled with a G protein. And these receptors activate a second messenger system that causes ultimately the opening of an ion channel. So if we look at the image below, an ionotropic receptor would be right here on the ion channel itself. The neurotransmitter would bind to that receptor, causing the opening of the channel. However, metabotropic receptors are not on the ion channel themselves. They're embedded in the cell membrane. So the neurotransmitter binds to a metabotropic receptor and kicks off a complicated process that's referred to as a second messenger system. And that complicated process plays out and ultimately causes the opening of the ion channel. Now we're prepared to talk about postsynaptic potentials. These are potentials not like action potentials that happen in the axons, but rather they're called postsynaptic potentials because they happen in the postsynaptic dendrites. As we mentioned previously, the action potential arrives at the axon terminal of the presynaptic neuron. It causes a neurotransmitter to undergo exocytosis, whereby it exits the cell and diffuses across the synaptic cleft to the postsynaptic dendrites. There it will bind to receptors, either ionotropic or metabotropic receptors, causing the opening of ion channels. In this example, we're again talking about the opening of sodium channels, which is going to allow positively charged sodium to follow its concentration and its voltage gradients to the inside of the cell. Since this is positive charge moving inside the cell, we can see that the inside of the cell will become more positive in charge. Therefore, it is called an excitatory postsynaptic potential, or an EPSP. Because it's letting positive charge into the cell, it's causing excitation. Because this is excitation in the dendrites, we call it an excitatory postsynaptic potential. But what about an inhibitory postsynaptic potential. IPSPs happen oftentimes with chloride channels. The neurotransmitter will come across the synaptic cleft, bind to chloride channels, causing those channels to open. And chloride is going to follow its concentration gradient to the inside of the cell. Since chloride is negatively charged, that means the cell is going to become more negative in its charge. By the way, there's actually an error in this image. This line should start down here at negative 65 millivolts and dip below, but just a slight error in the image. The point is that the cell becomes more negative. Rather than depolarizing toward excitation threshold as it does with an EPSP, this one's going to hyperpolarize away from excitation threshold. Hence, it is an IPSP, or an inhibitory postsynaptic potential. Now, there are actually two ways you can get an inhibitory postsynaptic potential. One is with transmitter binding to chloride channels, but you could also have transmitter bind to potassium channels, in which case the potassium channel would open and positively charged potassium would leave the cell. And remember, positive charge leaving the cell makes the inside of the cell become more negative. So you can either hyperpolarize a cell by allowing negatively charged chloride to flow into the cell, or by allowing positively charged potassium to flow out of the cell. So EPSPs and IPSPs. Now we can talk about summing the EPSPs and the IPSPs together. So summation can take place across two domains, a temporal domain and a spatial domain. What do we mean here? Well, if we take this target neuron, the question is, are we going to fire an action potential down the axon or not? Well, whether this neuron fires an action potential depends on how much excitation or excitatory input it receives versus how much inhibitory input does it receive. If this target neuron receives more excitatory input than inhibitory input, that will cause a net depolarization at the axon to excitation threshold. That will cause the opening of voltage-gated sodium channels, thereby kicking off an action potential down this axon. So we can sum these excitatory postsynaptic potentials temporally. That is, if we get a bunch of EPSPs coming together close in time, they will add together and depolarize the axon. But we also can have them coming from multiple spatial locations. So we can sum across space as well as across time. Of course, we also can sum inhibitory neurons. That's just summing negative signs, basically. The excitatory neurons would be plus signs. The inhibitory neurons would create negative minus signs. And if you have sufficiently more pluses than minuses, that will depolarize the neuron to excitation threshold. And that is temporal and spatial summation. And that basically wraps up our discussion of neural communication.